On this week's Arabidopsis Research Roundup, I'm delighted to be joined by Professor Mike Bevan from the John Innes Centre, and we're talking about a paper just published in Genes and Development. And it's got a long title. The title is Ubiquitylation activates a peptidase that promotes cleavage and destabilization of its activating E3 ligases and diverse growth regulatory proteins to limit cell proliferation in Arabidopsis. Okay, so thanks very much, Mike, for joining us today. And I'd say a long title, but it's quite an interesting and fundamentally important paper, I think. So it'd be great if you could give us an overview about it.
Well, thanks. The biological focus of the paper is part of our wider range of work that aims to understand how cell proliferation is controlled during organ formation.
Right.
Now, one of the basic, most fundamental observations in, for example, the growth of a leaf from a primordia is that cell proliferation occurs at an early stage of organ formation, and then it is suddenly it suddenly ceases, to be followed by a period of cell expansion and differentiation to form a normal-sized leaf.
Yeah. Okay.
And the same principles apply to the formation of petals and the formation of seed and teguments, which regulate is one of the main modes of regulating seed size. Mm-hmm. So we've been interested in this both from a fundamental point of view, because it is the genetic genetic analyses of growth control are really at a fairly early stage, both in plants and animals, and it's a fundamentally important aspect of biology. Now, we had some mutants in Arabidopsis that lead to bigger organs, bigger petals, bigger seeds, bigger leaves, and the plants take a longer time to grow to achieve their final size. These sorts of mutants, or phenotypes rather, are of strategic importance because they underlie variation in seed size and seed yield. One of the reasons we're following up this area of work is that it has potential applications in our crops of interest here at the JI, principally oilseed rape and wheat. Yeah. So we had these genes, and we had a lot of genetic interactions and yeast-2 hybrid interactions. What this paper describes is a biochemical mechanism whereby these genes, which are ubiquitin-regulated peptidases, influence the duration of cell proliferation during organ formation. We show first that the protein encoded by this gene family is a peptidase. Peptidases are pretty dramatic enzymes. They have a pretty much a one-way mechanism of action in which their substrates are cleaved. The protein substrates are cleaved. And that's pretty much an irreversible reaction. And peptidases are used in many, many regulatory systems in plants and animals, primarily characterized in animals, yeast, whereby peptidases are activated to enable a one-way transition.
Okay.
Perhaps the best described example is the anaphase promoting complex, where separin and separase are activated. So separase is a peptidase that then cleaves cohesin, and that stimulates the transition from anaphase to telophase. Mm-hmm. Now, so getting back to the mechanism, we showed that this peptidase is activated by ubiquitylation, which took us into a new and very interesting field. Mm-hmm. Proteins are modified by ubiquitin in many, many cases. Mm-hmm. The best understood case is polyubiquitylation that targets proteins for degradation. That's probably the best known example.
Yeah, absolutely.
But ubiquitin is a signal molecule that has got multiple functions. Both the numbers of ubiquitins added onto a substrate protein and the types of links between ubiquitin moieties have got very different signaling properties. We've shown that multiple monoubiquitylation, that is the addition of a single ubiquitin moiety at several sites on the protein, is sufficient to activate the peptidase. Mm-hmm. So we had a mechanism, and we also identified substrates of the peptidase. So looking downstream from the enzyme activity, so to speak, we identified first something that was paradoxical, and the reviewers found it fairly difficult to get their heads around,
right.
Maybe because we didn't explain it as clearly as we needed to. But the peptidase cleaves two E3 ligases that activate it.
Right. Okay.
Now, this sort of mechanism is in fact very common in ubiquitylation, and what it does, it creates a burst of activity. Mm-hmm. You activate something, and then you that activated enzyme then destroys the things that activate it. Mm-hmm. It's a well-known mechanism. Yeah. What that does, it creates a burst of peptidase activity. We identified other substrates of the peptidase by yeast-2 hybrid screens for interacting proteins. Several of these are regulators of cell proliferation. Mm-hmm. One of them is a histone deubiquitylase called UBP15. There are a couple of TCP3 so far, TCP transcription factors, and some other transcription factors that regulate opsin biosynthesis. So we have a bunch of diverse substrates, and these are cleaved by the peptidase. Mm-hmm. The idea is that the cleavage destroys their activity of the protein. Mm-hmm. So here we've got a model in which the peptidase is activated by ubiquitylation. It then goes on to cleave a bunch of regulators of cell proliferation, positive regulators of cell proliferation. Mm-hmm. So that the model that we're testing now is that the peptidase is involved in determining the transition from cell proliferation to cell expansion and differentiation by the concerted cleavage of regulatory proteins. So what I find interesting about this work, of course, I would find it interesting. It's not a self-serving comment. It's one of the first examples that we have of the regulation of the proteome during growth and development. Many studies, of course, interesting studies, show how transcription is regulated to control differentiation mm-hmm. and growth. But there are very few examples, particularly in plants, of how the proteome is regulated. So this took us into contact with a group in Germany, Nico Dismayer's group, and he's an expert on the end rule degradation pathway. Yep. So we asked the question, well, once these things are cleaved, what's the fate of the cleaved protein? And Nico was able to show in our collaboration that once the proteins are cleaved, this reveals a new N-terminus that is then the target of another set of E3 ligases that polyubiquitylate those proteins and lead to its degradation by the proteasome. Mm-hmm. So that's the current state of the work. That's what the paper describes. Our current work is focused on at least three areas. First, we're trying to identify other substrates of the peptidase by getting knowledge of its cleavage site specificity. Mm-hmm. Okay. That's going reasonably well, but it's fairly slow work. Another area of work is identifying how these proteins work in wheat and oilseed rape, right. In particular wheat. So we have started a program to do some CRISPR in wheat to knock out the gene families this being complemented by biochemistry, we can show that the wheat equivalents function as E3 ligases that can control the wheat peptidase.
Mm-hmm.
Interestingly, natural variation, it's more than natural variation. There is genetic variation in wheat that has been selected by breeders for increased 1,000 seed weight. And that is genetic variation in one of the components of this system. So we're busy following that up because this QTL in wheat gives a 5% yield increase in good seasons. There you can sort of see it's a John Innes type activity. We study mechanisms in Arabidopsis and do our best to see if that's relevant to crop plants. The other area of work that we're working on is what are the upstream regulators of the peptidase? So we know they're triggered by ubiquitylation by some E3 ligases, but what makes them start to be active as negative regulators of cell proliferation? So we're again using the power of Arabidopsis. We've got an interesting candidate mechanism that we're looking at. And finally, we are interested because the ubiquitin activated peptidases influence the transition from cell proliferation to cell expansion during leaf growth. We are using live cell imaging to measure cell proliferation in real time during leaf growth in mutants and mutant plants. This will give us a precise measure of their effect on cell proliferation and growth. It'll tell us where they're active and when they're active in the growing leaf. So in summary, that's what the paper's about.
Yeah, no, that's excellent. So you've really linked all the pieces together there. And just to touch on that final point, it'll be very interesting to see where the activity of this peptidase then links into this endoreduplication and the doubling of the amount of DNA, which allows the leaf then to get to that largest phase. And I'm sure you're looking into that sort of thing.
We'll be able to test endoreduplication because in this imaging system, we will be building some fluorescent proteins that mark nuclei. You can tell how big the nucleus is, and that will tell you how its ploidy level. We think that the peptidase is primarily involved in limiting proliferation. It cleaves TCP15, which has got a role in mediating the activities of retinoblastoma and a cyclin.
Right.
And the balance of those activities regulates whether you undergo a mitotic cycle or an endoreduplication cycle.
Okay.
We're not getting that. We're not experts in that field. Hopefully, others can pick up on this.
Yeah, absolutely.
Experts in the cell cycle can work on it, but we're not going to sort of burrow down too much into how it influences mechanisms in great detail.
It just reminded me, really, this is a little advertising for GARNet, in that there's a paper a couple of weeks ago that I flagged up on the GARNet blog about a system of endoreduplication in the sepals of Arabidopsis flowers. This fantastic system where there's these enormous cells and the signaling which transitions them to one cell fate or another. Anyway, so it's very interesting.
I must read that.
Please take a look, indeed. So one final question. So you mentioned this about the targets of the peptidase. Are there conserved motifs that this peptidase cleaves? Have you identified those motifs, or is that something for the future?
We have identified the motif in 3 cases.
Okay.
And it is a sort of a suggestive motif, but it's not. The peptidases have a preference for just a couple of amino acids at their cleavage site. Ours has got a preference for a hydrophobic amino acid followed by a basic amino acid on one side of the cleavage site, and an asparagine, the new C-terminal site of the peptidase cleavage site. So this is sort of enough to guide you, but I wouldn't want to use it in a screen of the entire protein.
Yeah, okay.
We'd reduce the candidates in Arabidopsis from maybe 25,000 down to about 10,000.
Right. Okay.
Yes. And that's not a good starting point.
There's a little more complexity there than that. That's for sure. Lovely. All right. Well, thank you very much, Mike. You've really done it's been a very nice description, and as you say, linking in the strategic goals of the JIC and with this nice biochemical and genetic study as well.
I've really enjoyed the biochemistry. I was trained as a biochemist, and it has been marvelous to get back to it. But biochemistry in the context of genetics and
absolutely.
development.